This lecture looks at the general and special senses. However, in particular, we are going to focus mostly on the special senses. And the reason for that is because we have covered the general senses to a pretty solid degree throughout the nervous system in the 5 chapters that we cover there. So the first thing we want to talk about is the difference between sensory information, i.e. stimulus, and then a sensation. A sensation is what we determine to be conscious awareness of incoming sensory information. So basically any stimulus for which you become consciously aware of, which you think about consciously, that is considered a sensation. Whereas a stimulus is merely information that has come in through the peripheral nervous system that has been detected by a receptor. And it doesn't become a sensation until it actually is, you're consciously aware of it and it's dealt with in your cerebral cortex. Now, as always, stimuli are still detected by receptors, and we've talked a great deal about this in the nervous system. Now, there are 2 classes of senses. The first part, those general senses, temperature, pain, touch, stretch, pressure. These we've talked about quite a bit more broadly in the nervous system. But the ones we really have not talked about are the special senses: gustation, olfaction, vision, equilibrium, and then finally audition. Now, receptors can range in complexity from a single-celled dendritic ending of a neuron to a complex sense organ. So it can be a dendritic ending of a neuron, or it can be something as complex as the eye, or the inner ear, or the taste buds on the tongue. Now, the idea behind receptors is that they monitor both internal and external conditions and conduct information about the stimuli that they're collecting to the central nervous system. And one of the ideas that we want to kind of drive home about receptors is that there's an inverse relationship between the size of the receptive field and our ability to identify the exact location that stimulus came from. So if the receptive field is small, that means that precise localization and sensitivity are very easily determined. So that is that the precision is great. So if the field is small, the precision is great, that we can determine exactly where that sensation or that stimulus came from. However, when you have a very broad receptive field, then you often have only a general ability to determine where that exact stimulus came from. So it's a bit more difficult to precisely determine the location of the initial stimulus. Now, we have a couple of different sensory receptors, and we want to talk a bit about the idea of acclimation. So tonic receptors are involved in maintaining our balance. They allow us to help keep our head upright. So they're the receptors that if we lean too far back in a chair, that we feel that sensation that we're falling backward, and then we obviously respond to it by trying to make sure we right ourselves before we fall over. Phasic receptors are different. These are things that per se signal the increased pressure on our skin. So think about the idea of pinching somebody or pinching yourself, right? That information is taken in through a phasic receptor. And phasic receptors can undergo acclimation, which is where we have an active reduction in the sensitivity to a continually applied stimulus. So it's the idea that you can become less and less sensitive to a stimulus as it becomes more and more frequent. Because, again, if it's more and more frequent, it's less likely to become a detriment to our survival, or it's easier to ignore. There could be a lot of reasons why we acclimate to it. When it comes to receptors, it depends upon where we find them. Special sense receptors are housed in those complex organs in the head, so the complex sense organs. Whereas general sense receptors are just distributed throughout the skin and organs, so they are continuous to a great extent with our idea of the peripheral nervous system. And we classify these based upon location of the stimulus or their location on the body. Exteroreceptors are receptors that are found in the skin and mucous membranes and are open to the outside. And these include our special senses. Interoreceptors, these are receptors located within the walls of our viscera and are monitoring sort of the internal environment within our body. Proprioceptors, these are receptors in our skeletal muscles, tendons, and joint capsules, and help us understand our relative position of our limbs and whatnot, the stretch in our skeletal muscles, the extension and whatnot in our joint capsules. We can refer to these by body location as well by thinking about what they do. So chemical receptors per se are obviously going to respond to specific molecules in fluid. Temperature responds to changes in temperature. Pain is pain, detects damage to tissue. Touch detects fine or light touches, and this is something that you want to make sure you discern from pressure. Pressure responds to mechanical pressure, vibration, and stretch. So pressure and touch are different things. The idea of touch I usually like to use when we think of if you've ever had a sibling, if you ever as a kid messed with one of your siblings, and they would fall asleep, and you would tickle the hairs on their arm or on the back of their neck or something. That is fine touch. That is light touch. Pressure is when you press on something. That's pressure, right? Proprioception, we just talked about change in position, tension, stretch of a muscle or joint. All right, so let's first start talking about taste. This is referred to as gustation, and we find this entirely on the dorsal surface of the tongue. And the gustatory receptors are housed in specialized groupings of cells that we call taste buds. And taste buds lie along the sides of epithelial projections that we often refer to as papillae. And here again is this term that we see once again being used, the term papillae. Each bud contains about 40 gustatory cells. This works in combination with olfaction, and I think all of us understand that from being sick at one point in our life. You had a cold that carried with it a great deal of congestion. You would have recognized pretty quickly that if you can't smell very well with your nose, then all of a sudden you can't really taste very well in your mouth either. And so you have that combinatorial effect. There are 4 specific types of papillae that we're going to cover here. The first are called filiform papillae. These are found on the anterior 2/3 of the tongue. These do not house taste buds and have no sensory role. They're more for friction and for drawing things into the mouth. Fungiform papillae are primarily on the tip and the sides of the tongue, and these contain only a few taste buds each. Veilate, or sometimes referred to as circumvallate papillae, are arranged in an inverted V on the posterior dorsal surface. Now, the interesting thing about veilate papillae is that they are the least number in terms of papillae. They're least numerous in terms of the number of papillae, but they have by far the highest number of taste buds per papillae. And each is surrounded by a deep, narrow depression. Last papilla is going to be the foliate papillae, and these extend as ridges on the posterior lateral sides of the tongue. Now, these house only a few taste buds during childhood, not well developed overall on the human tongue. Now, there are 5 basic taste sensations, arguably the potential for 6, but we're going to cover the 5 today. So there's salty, right? The taste that we associate with metal ions. This is a salty taste. There is sweet taste, and this is often that taste that we associate with organic compounds like sugars, like glucose. There is the sour taste that we associate with acids. So this being something of a low pH, something less than 7. Whereas there is a bitter taste. This is the taste association with alkaloids, and an alkaloid is a molecule like Tylenol, for example, with a pH of 7 or of above 7. And if you ever have had Tylenol on your tongue, for those of you that have had a Tylenol on your tongue that's not coated, that pill is extremely bitter, and it's often leaves a very unpleasant taste in your mouth because of that bitterness. That's because it's an alkaloid, and we often respond to alkaloids as being much more toxic or dangerous. Umami, I love this flavor, I love this sensation. Umami, this is sort of that savory flavor, that savory taste. And this detects amino acids, and it's a pleasant taste. Think of like chicken soup or vegetable broth. It's a broth flavor it's often associated with things like that. And that's because you're picking up on the amino acids, and this in turn is a pleasant taste to the body. Now, there's arguably a 6th taste. However, this one is not necessary and known for sure yet, but that is the taste of water. And it's thought that we might be able to taste water in the pharynx, but again, that is not yet known for sure. Now, olfaction. So this is the sense of smell. The receptor cells here are used to detect odors. Now, these are bipolar neurons that synapse with the olfactory bulbs. So these are olfactory bulbs on the actual brain. So these would be cranial nerves 1. The supporting cells are these cells. So this is that bipolar neuron that I was talking about, and here are the olfactory hairs. These are actually the receptors here. And the large orange or pink cells between those, these are called supporting cells. And these are cells that sort of sandwich these olfactory nerves. They help sustain and maintain the receptors, so they're there to help provide space and protection and cushion for these very important neurons. The basal cells, on the other hand, these are kind of spread throughout, but you see them here in purple. These are stem cells, and these are there to replace olfactory epithelium, which is this tissue overall, when, as it turns out, you damage it. And that does happen from time to time. For those of you that have ever opened the oven and not thought about it and just took a smell because something smelled really good, but then you forget that the heat, the initial wave of air that's coming out of that oven is going to be incredibly hot, and so you burn the inside of your nose. That can often initially make smell a lot harder to detect. Now, the olfactory system in humans is kind of pathetic, but we can recognize as many as 50 or 60 different primary odors, whereas dogs can distinguish thousands of these different primary odors. So ours is more basic or basal level in terms of its abilities. The epithelium itself covers the inferior surface of the cribriform plate. So here is that cribriform plate from the ethmoid bone, everybody's favorite bone. And these are the cribriform foramina through which those neurons are passing. And that epithelium in this area here also covers things like the nasal concha and the nasal septum. And the glands within this epithelium secrete a thick pigmented mucus. Vision is the ability to visually perceive light, color, and movement. So we have visual receptors, what we call photoreceptors, that allow us to actually discern from these things. And so the visual receptor itself is actually what allows for the sense of vision. But there are plenty of accessory structures on the eye that help the eye do its job, but are not directly involved in the ability to detect light, color, and movement. So some of these are things like the conjunctiva. The conjunctiva is an outer covering on the front of the eye that covers the cornea and a bit of the anterior portion of the sclera. So it's that anterior exposed surface. Now, all of us have had some kind of run-in with the conjunctiva. We just don't think about it. If you've ever had pink eye is also known as conjunctivitis. And conjunctivitis is an inflammation and infection of the conjunctiva. And by this respect, you often feel that scratchy, itchy feeling in the front of the eye, and then the overproduction of that almost thick fluid that actually ends up gluing our eye shut if you let it go for too long. That is kind of characteristic with pink eye or conjunctivitis. The eyebrows, eyelashes, and eyelids obviously do not allow you to perceive light, but yet are not any less important. They prevent foreign objects from coming into the eye or coming into contact with the eye. They're there to help keep things away. Obviously, the eyebrows and eyelashes are there to help keep, say, dirt, if you have dirt on your forehead or dust on your forehead and you're sweating. It helps keep that dirt from flowing down with the sweat into your eye. The eyelids are obviously there to help keep something from coming into the eye. So, for example, if you're walking into a dust cloud or driving into a dust cloud, say you're driving and your window's open, and you don't have enough time to actually close your window, sometimes you might close one or both eyes to keep the dusty air from getting in there. The lacrimal glands are there to keep the eyes moist, keep the exposed surface of the eyes moist, clean and lubricated. So the lacrimal glands are the same glands that allow you to cry. And the fluid that they produce has a great deal of dissolved molecules in them that help keep it just generally moist, but also help keep it clean, help keep it from getting infected. And then finally, we have the extraocular muscles. And the extraocular muscles are there to actually move the eye so that we can look up or down or left or right or straight ahead. Just going over this real quick here, the conjunctiva is going to line this inner surface here all the way along to the cornea and then back here. So that's that conjunctiva. Eyebrows, eyelids, obviously, and the eyelashes. We've covered that material so far. Now, to also make clear some of the other structures that we should find here, the cornea is that big dome window, that clear dome window in the front here. So that's the part of our eye that we're most often used to or associated with it. The white of our eye, that white layer that goes around almost the entirety of our eye, minus a little small portion in the back, and then the area that takes up the cornea, is called the sclera. That is another tunic of our eye. And the sclera and the cornea are actually continuous with one another. So where the sclera ends here, the cornea picks up so that we have a clear window to the front of our eye. And then here where the cornea ends, the sclera will again pick back up again. The choroid is this layer just deep to the actual sclera. And this is a vascular layer, so there's actual vasculation. There's blood flow in this layer. And then finally we have the deepest most layer here, the actual retina. We see the retina here in yellow all the way, covering almost the entirety of the eye except for the front of the eye and this little portion in the back here. Other structures that are of note that we'll talk about in lab are things like the iris. This is that structure here. It's often colored or pigmented. The space in between the portions of the iris, this is the pupil. This large structure here that is cross-sectioned is the lens. These white ligaments here are the suspensory ligaments. This structure here, we can see the ora serrata here, and we can see the ciliary body. So the ora serrata is this posterior-most edge. It looks like a toothed or serrated margin of the ciliary bodies overall, which are this whole margin of structures right here. Now, if we look back to the very posterior aspect of the eye, we'll see what we call the optic disc. And this is the area where there's a gap between this portion of the retina here and this portion of the retina here. This gap lacks any photoreception whatsoever, and it's considered to be the blind spot. So optic disc and blind spot often go hand in hand in terms of the name being used. And the idea is that it's called the blind spot because there's no photoreceptors and thus no image forms there. Now, here's the interesting thing. So this area here is actually the blind spot, and this little area here is called the macula lutea, called the yellow spot. And this area is a depression, and it's a rounded yellowish region. It's a pit, and the pit deep within it is called the fovea centralis. And this is the area of sharpest vision on the retina. And by sharpest, I mean it contains the highest proportion of cones and almost no rods. Now, the cavities and chambers of the eye are a bit confusing, so try to follow along with me here just because it's kind of a pain. So, first I want to take the eye and want to split it up into its two cavities. And so we see that associated with the actual lens. So, here's that lens again. It's a cross-section of the lens. So this anterior surface of the lens, all the way forward to the posterior aspect of the cornea here, this area from here to here is considered to be the anterior cavity. And the posterior cavity is from the posterior surface of the actual lens all the way back to the anterior surface of the retina. And in the anterior cavity is a fluid that we call the aqueous humor. This is a very liquid-based filling fluid that occupies these areas. And in the posterior cavity, this fluid that is within this chamber is actually, within this cavity I should say, is actually much more like a stiff jelly, and it's called the vitreous humor. Now, let's break down the anterior cavity a little bit more into its two separate chambers, because the anterior cavity has actually two legitimately different spaces. So from the front, or that is the anterior surface of the lens, to the posterior surface of the iris here, this is called the posterior chamber. And then from the anterior surface of the actual iris here to the posterior surface of the cornea, this is now the anterior chamber. So we've got two chambers within the anterior cavity. The posterior cavity, however, is just that. It's just the posterior cavity. No chambers within it. Now the humor. I'm going to talk about these two because they are different. The vitreous humor is very thick. It's very gelatinous. It's there to help the eye maintain its shape. And that again is seen in the posterior cavity. The aqueous humor, however, this is aqua, water, fluid. So it's at the very much the same consistency of water. And this is meant to flush waste products and remove them from the eye. It's very fluid as a result. The aqueous humor is secreted into the posterior chamber. It flows around the lens, through the pupil, and into the anterior chamber. So it's going to flow in this direction. And then it's going to flow outward. These structures here, called the canal of Schlemm. This is the old term, canals of Schlemm. One of my favorite terms ever in anatomy is the canals of Schlemm. Now they're called the scleral venous sinuses. But before, they were called the canals of Schlemm. Either way, it's small sinuses on the eye that are help meant to remove fluid from the eye and allow it to drain. Now, as we talked about before, when it comes to visual pathways, we see decussation that exists in both eyes, so both the left eye and the right eye, whereby information from the left eye goes to both the left and the right hemisphere, and vice versa with the right eye. Now, at the optic chiasm, some of those axons are actually going to decussate, and we see that here, left eye being all blue axons. Some go to the right, some go to the left, and same thing with the right. They're all in red axons. Some are going to go to the left, some are going to go to the right. So in essence, the optic tract, which is either here or here, contains axons from both eyes. And visual stimuli is going to be processed by the hypothalamus and then interpreted by the visual association areas of the cerebrum. We see those back here at the occipital lobes. Hearing equilibrium. So we're going to move past the eye now and into our ability to hear, that's audition, and then equilibrium, which is our ability to maintain balance and know where we are in the world in terms of are we upside down, right side up, or whatnot, compared to the way we're supposed to be. Now, the external ear itself is this area here, and this is largely just meant to be a funnel. This is located mostly outside the body, and the middle and inner areas, middle here and then the inner here, are housed within the petrous portion of the temporal bone. So for those of you that remember the petrous portion of the temporal bone, that is in fact the middle and inner ear. Now, sound is meant to be collected here by the auricle and then funneled through the external auditory canals onto the actual tympanic membrane. And the idea here is to convert these sound waves into mechanical movement. And this whole system is based upon movements in the inner ear fluid. So sound waves will come in this direction, so through the external auditory canal, they'll vibrate the actual tympanic membrane, which will in turn vibrate each of the three inner ear ossicles. Right here we have the malleus, the incus, and then finally the stapes that is attached to the inner ear. And that will then register movements. Those vibrations will then in turn register through the actual inner ear, and the changes to the fluid inside the chambers within that inner ear are actually what is then sent off to the brain for interpretation. So those are the sensations. Okay, to go through these things again, the external auditory canal or meatus is this region right here. The auricle is the overall ear on the flap on the outside of the skull right here, that soft tissue portion of the ear that we often think of being the ear. Now, we have a helix and an antihelix, a tragus and an antitragus, none of which are seen really great here. You'll see them much better on the models in the lab. The tympanic membrane, I've referred to already once or twice, is this white membrane here that is meant to register those sound waves coming through the external auditory canal. And the function of this system overall is to funnel sounds this direction. The middle ear contains an air-filled tympanic cavity, so this whole area here is all filled with air, which is why we feel such changes in pressure on an airplane as we're climbing in altitude. Now you've got a bony wall all the way around this structure here, almost all the way around. But we do maintain an open passageway, an open connection to the atmosphere through the auditory tube, which connects to the pharynx here, nasopharynx specifically. And we call this the auditory tube or the eustachian tube. And so the reason why you can drink fluids or chew or swallow during ascending in atmosphere within a plane and help regulate that pressure in your ear and make your ears pop is because of this open passageway here, the auditory tube, right here. Now, the middle ear is what houses the auditory ossicles, the malleus here, the incus here, and then finally the stapes here. Now, the eustachian tube is that open connection into the nasopharynx that we see all the way here. And it's that air fluid or air movement through this tube that results from the following movements that allows pressure to equalize on both sides of the tympanic membrane. So that way, when we increase in altitude, the pressure doesn't actually burst our tympanic membrane. Now, within the inner ear, there's a lot of structures here. Not all of them we're going to cover all that particularly well or in any kind of detail. What I do want you to recognize is that all these passageways in here are actually fluid-filled chambers or tubes. And we've got a couple of different labyrinths within this system. We've got the membranous labyrinth here. You can see it surrounded by a pink membrane. And within that, we've got a fluid that we call the endolymph. And then you've got all these blue lymph areas and the actual spaces within them. And these are called the bony labyrinths. And the blue fluid within them is actually called paralymph. The major area where initially sound comes into, or sound, I should say, vibrations come into, is called the vestibule. And this information is then going to pass into the actual cochlea we see here for interpretation. The semicircular canals here, here, and here are kind of a three-dimensional axis, and they allow us to deal with equilibrium. So this is that equilibrium system for the special senses. And the idea that I want you to keep track of is that if you look here in this slide, you see these purple extensions of a cell that we call the hair cell. And they're sticking out into the fluid. And we notice that when the woman has her head upright like this, like any of us would, the fluid is held in a certain position, and thereby so are the actual hair cells. And this is interpreted by the body as a upright head. Whereas when she tilts her head forward, or downward I should say, then gravity takes hold of this fluid and pulls the fluid downward, and thereby we see a response by the hair cells in kind. These hair cells actually begin to fold forward as well. And that response this stimulus is going to be interpreted by our brain as our body or our head tilting downward and forward, and then we have to do something about that, potentially. Now, equilibrium, just as we were talking about here, is rotation of the head that causes the endolymph within the circular canals to push against a structure we call the cupula, which is covering the hair cells. And so this is that cupula. And when it moves, the hair cells move. And when the hair cells move, this in turn then registers as a certain stimulus to the actual brain.